what we ended up doing, we designed a 10,000 bead Infinium tomato array. So what this is an array that Illumina designs that has 10,000 assays on it. And this is the breakdown of the assays. You can download all these slides off the internet, so you don't have to write all the numbers down. So the SNPs from cultivated germplasm from the SoulCap project of the, actually I'll explain the difference, 8,700 that are actually on the array. 4,500 of them are from the SoulCap project. We actually worked with a company called Trait Genetics run by Martin Genau in Germany. He's a service company that also has developed SNPs in tomato and validated SNPs. He gave us about 1,500 sequences to work with, and we compared those with ours. We also got about 400 sequences from Mathilde Kaus, who works with INRA and Avignon on fresh market tomato for most part. And we put those all in the pot and we looked for the non-redundant set. And that's what the rest look at. So when I say community, these are SNPs that were found by at least two of the groups, either us and INRA, us, in Germany, or any other combination. 1,300 were found by other groups, as a group overall. 500 were associated with genes, so these candidate genes. And with the rest, we tried to cover the genome the best we can. And because we don't have a genetic map for the SNPs, we actually have a genome, and we know the sequence. We can blast those sequences back to the genome. The genome is ordered by chromosome in the physical map, and if we try to have a SNP in each scaffold, this kind of higher level assembly, we have a good representation of the genome. And we know our SNPs are going to be spread out through the genome, even though they're not directly genetically mapped. So what's nice about something like that, we don't need polymorphism. There are SNPs between the varieties we actually sequence, but we just know the sequence that those SNPs are based on mapped to these places in the genome. So they're automatically bioinformatically mapped. So in the end, we end up with 8,784 actual SNPs that are made up of this. The trick about the Infinium assay is this: if you have a transversion SNP, so an A to a T or a G to a C, it needs 2 spots on the array. So you're using twice the hardware in a sense. So you have 10,000 spots, you have to decide how you fill them. And so for 1,200 of the SNPs, they're AT or GC SNPs, transversion SNPs. So there we had to use 2 spots. So we had to have 1,200. So even though we have 10,000 spots, we only really have 8,700 and 84 assays on that. And so those chips are actually being physically manufactured right now. We don't expect them, hopefully some of you have ordered them in the audience. We had a consortium, about 2,000 samples were ordered and from Illumina, and they're available really now to anybody to order. But they won't be available, I think, physically till about December is when the first ones will come out. And that's when we'll get them as well. So we won't know how well they work till then either. But based on our 96 data, which is the exact same type of filters that we use, we're pretty confident that these will work quite well. And then I can't thank John Hamilton, Robin Buell, and David put in a lot of time to try to get this data set refined into something that we think is going to be useful for the tomato community.